Hi, I'm Anida, and my members are Jinwoo, Chuanqi, Arifa, and Shujin. And we are from Group 12, and we're going to present about enzyme kinetics and modeling of enzymatic systems through a case study. In this presentation, we're going to introduce on enzyme kinetics through a case study, after which we shall talk about Michaelis-Menten kinetics as well as the significance of Km and Vmax, and finally we'll go through the data analysis of the Km and Vmax before ending off with the conclusion. So let's look at the case study here. In this case study by the Biotech Instruments Company, an absorbance-based assay was used to study the enzyme kinetics of beta-galactosidase with O-nitrophenol beta-D-galactoside, or ONPG, as the substrate. A data analysis software called Gen5 was used to obtain the Michaelis-Menten equations and the Km and Vmax. The effects of phenylethyl beta-D-thiogalactopyranoside, or PETG, on beta-galactosidase as an inhibitor were also studied. Beta-galactosidase is an enzyme produced from the LEGZAC gene, which can be found in the bacterial cells. Beta-galactosidase catalyzes the hydrolysis of the terminal non-reducing residues of beta-D-galactosides, such as lactose, ONPG, or XGAL. ONPG is a synthetic compound that forms O-nitrophenol, or ONP, and galactose when hydrolyzed by beta-galactosidase as shown in the figure below. ONP is a yellow compound that absorbs light at 420 nanometers wavelength, which is useful to monitor the enzyme activity through an absorbance-based assay. To study the enzyme kinetics of beta-galactosidase, ONPG substrate stock solution was diluted with a reaction buffer and added into microplate wells. Beta-galactosidase was also added to initiate the reaction with the substrate ONPG. To study the effect of PETG inhibitor, dilutions of PETG were also added into equivalent beta-galactosidase reactions. Beta-galactosidase activity is measured and analyzed through an absorbance-based assay using PowerWave HT microplate spectrophotometer and Gen5 data analysis software.
In this case study, the application of Michaelis-Menten kinetics was utilized to study the kinetic analysis of beta-galactosidase activity. The Michaelis-Menten kinetics is a model of enzyme kinetics which explains how the rate of an enzyme catalyzed reaction is affected by the changes in substrate concentration. The model helps us to understand how the bacterial enzyme beta-galactosidase interacts with the O-nitrophenol beta-D-galactoside, in short form, the ONPG substrate. In Figure 2, when the Michaelis-Menten graph of the reaction's velocity against the ONPG substrate concentration is plotted, the rate of the reaction initially exhibits a rapid linear increase as the substrate concentration increases, and eventually reaches a plateau, where further increases in the substrate concentration have no effect on reaction velocity, as all enzyme active sites have been saturated with the substrate. The graph is a visual representation of the Michaelis-Menten equation, where the equation relates to the maximum rate of the reaction, Vmax, Michaelis constant, Km, and the substrate concentration. These kinetic parameters of Vmax and Km will be explained further in the next slides. The Michaelis constant describes the affinity that the enzyme has for its substrate. It is also known as the substrate concentration that results in 50% of the enzyme velocity. In this case study, the significance of Km is to determine how efficient beta-galactosidase at catalyzing the hydrolysis of beta-D-galactosidase into sugars.
Based on the case study, there's two different methods that can be used to determine the Km, and firstly, it is using the Lineweaver plot, and the second method is using the Gen5 software. As shown in figure 3 on the left, it is plotted using experimental data using the reciprocal of enzymatic velocity against the reciprocal of the substrate concentration. Using the equation shown here, the Km value can be calculated based on the slope of the plot, and the slope is determined by the Km divided by Vmax, as shown in the equation here. In the case study, the Km value is calculated as 0.3449 millimolar. When we're using the Gen 5 software, it is actually a software that is developed for imaging and microscopy applications. It is user-friendly and provides a detailed analysis of the data that we will be discussing. So if you were to look at figure 4, and given that based on Arifa sharing, Km is the substrate concentration at 50% of Vmax, and we'll be using that to deduce the Km that is shown in figure 4. This would leave us with a value of 0.24 millimolar based on what was calculated in the case study. And if you were to compare these two methods, the Gen 5 software would be considered more accurate because it is using a computer software that takes into account other factors as well. Figure 5 is also plotted using the experimental data, but the main difference between figures 3 and 5 would be that figure 5 focuses on the effects that the PTG inhibitor has on Vmax and Km. So just for additional knowledge, PETG stands for phenyl ethyl beta D-deogalactopyranoside. As you can see in figure 5, the blue line was plotted based on a sample that has a high concentration of PETG inhibitor. This led to a significant reduction in the enzymatic activity, and as such, the enzyme would have a very low affinity for the substrate, and a high Km value would be produced. This value is evident in the steep gradient of the blue line, as you can see here. On the other hand, the black line was plotted based on a sample that consists of a low concentration of PETG inhibitor, and this led to the enzyme having a high affinity for the substrate, since there is no more inhibitors to compete with the enzyme for the active sites. This would produce a low Km value, which is evident in the gentle gradient of the black line.
So I'll be talking about maximum velocity, known as Vmax. Vmax is the maximum reaction velocity at which all enzymes become saturated with the substrate in an enzymatic reaction. In this case study, the significance of finding Vmax is to determine the maximum velocity of enzymatic reaction when beta-galactose is saturated with substrate OMPG. To determine Vmax, Lineweaver-Burk graph was used. As mentioned in slide 9, the y-intercept represents the reciprocal of Vmax and found to be 0.039. The case study also utilized Gen 5 software to simplify the routine data analysis, and hence, based on figure 7, Vmax was estimated to be 33.4 milliOD per minute. To test for the inhibitor effect on hydrolysis, the experiment was conducted with and without the inhibitor. Two different values of Vmax were obtained and compared. And it was found that both experiments achieved similar Vmax values. As seen in figure 8 and 9, we can see that both PETG and OMPG contain galactosides that are circled in red. This implies that they have similar molecular structure and hence will have similar enzymatic reaction. In figure 10, it can be seen that the green line represents the highest inhibitor concentration and the black represents lowest inhibitor concentration. This shows that the gentler the slope, the more effect that PETG inhibitor has it on the substrate. As mentioned from the previous slide, since PETG has a similar chemical structure as OMPG, this suggests that it will bind to the same active binding site of the beta-galactosidase enzyme. This further shows that PETG is a competitive inhibitor as it will compete for enzyme binding sites, leading to an excess of substrate available. Hence, this results in similar Vmax value in enzyme reaction with or without inhibitor present. As seen in figure 11, Gen 5 software also shown that the greater the inhibitor concentration, the lower Vmax reach.
To wrap up the presentation, here's a summary of what we have discussed earlier. First of all, 2 experiments were conducted as per experimental case study. The effect on kinetic enzymatic activity of beta-galactosidase was investigated while varying concentrations of substrate OMPG as well as inhibitor PETG, which has a similar structure to OMPG. For both experiments, it was proven that once the enzyme was saturated with either substrate or competitive inhibitor, the Vmax would be reached. Next, we explored the Michaelis-Menten kinetics, which essentially sums up the relationship between Km and Vmax with respect to the substrate concentrations. These 2 parameters are important because it allows us to study the effect on enzyme kinetics that can provide useful insights for various reaction mechanisms, hence allowing the optimization of bioprocesses. Lastly, to recap, Km and Vmax can be obtained either manually using Lineweaver plot or digitally via Gen 5 data analysis software. However, between these 2 methods, Gen 5 software is preferred due to higher accuracy, efficiency, and flexibility in manipulating data. It also reduces potential errors associated with manual calculations. And that's the end of our presentation. Thank you.